Hello, my name is Véronique Karsten. As Product Manager of Molecular Biology Products at MP Biomedicals, I'm pleased today to give this talk targeting microbiota. The aim is to share with you a comprehensive solution to enhance microbial world exploration with a proven standardized sample preparation. The outline of the presentation highlights 6 objectives of microbiome research applications. The first step is to lead microbiome research forward using molecular methods. Second goal is to select optimal tools for unbiased microbial cell lysis. The next step of the workflow is then to achieve high-quality metagenomics DNA extraction. Another objective is to obtain pure RNA for metatranscriptomic analysis. The fifth section of the presentation is dedicated to protocols to co-extract DNA and RNA for combined applications. And the last part of this talk will focus on automated DNA and RNA purification solutions. Microorganisms building a microbiome are composed of bacteria, fungi, viruses, archaea, and protozoa. These fascinating microbial environments are found everywhere in all parts of the Earth's biosphere. Major environments hosting microbiome communities are human and animal bodies, soil, oceans, and plant specimens. Today, only 1% of the microbiome has been researched so far. Today's research goal is getting to know the trillions of bacteria and other microbes in, on, and around us. Research on microbiomes showed that these microorganisms are intrinsic to our health, and microbiomes are also fundamental to key geochemical processes. Human microbiomes synthesize vitamins and neurochemicals. They aid digestion, strengthen the immune system, influence processes in the brain, and play a role in certain cancers. Soil microbiome is involved in the cycling of nutrients and provide plant protection against biotic and abiotic stress. And ocean microbial inhabitants are also called nanobiome. They produce half of the oxygen we breathe and recycle the same proportion of carbon dioxide from the atmosphere. 4 research segments dedicated to microbiome research are defined according to the microbiome habitat. The human health segment is the first one. The human microbiome fulfills functions essential to life. A microbial imbalance, also called dysbiosis, induces diseases. Microbiome research studies explore the development of therapeutic tools. The second segment is the environmental segment. The microbiome plays a fundamental role in ecosystem processes essential for all forms of life on Earth. Microbiome research projects focus on understanding and meeting the challenges of environmental changes. The agriculture and food segment. The microbiome plays a role in plant growth and disease resistance. Microbiome research approaches explore new ways to improve agriculture and food production. And the biotechnology research segment, in which researchers are investigating how microbial communities could be used more effectively to convert waste into biofuel and bioenergy and treat wastewater. What are the most common starting sample types used in the study of microbiome? Feces, Mucosal biopsies and skin are commonly used to study human and animal microbial communities. Soil, sediment, sludge, compost and water are examples of starting samples of environmental analysis of the microbiome. Rhizosphere, roots, wood, vegetables, and food produce are used in agriculture and food research labs. Sewage and industrial waste are commonly used for a wide range of industrial and environmental biotechnologies applications. Several technologies are used to explore microbiomes. The two major approaches for analyzing microbiomes are 16S rRNA gene sequencing, technique that allows the identification and quantitation of microorganisms, and shotgun metagenomic analysis that is conducted to enhance the knowledge on the taxonomic and functional profile of a microbial community. In parallel, metatranscriptomics allow to obtain whole gene expression profiling of complex microbial communities. These molecular biology technologies always start with the same experimental workflow. Sample collection, followed by sample disruption and cell lysis. DNA is then isolated from the lysate and purified for 16S rRNA diversity analysis or metagenomic analysis. RNA is extracted and purified for RNA expression profiling. Microbiome research projects require an expertise in dealing with challenging samples. Sample preparation is a critical point. An inefficient method can have a big impact on the microbial community analysis and lead to biased results. An insufficient sample grinding step will affect the lysis efficiency of the microorganisms included in this sample and therefore skew results. An inefficient cell lysis procedure that selectively breaks open easy-to-lyse microorganisms while difficult-to-lyse cells like spores and gram-positive bacteria, for example, will not be lysed, will affect the results of the microbial community analysis. Sample prep methods leading to low yields of DNA, poor DNA quality, or co-purifying substances coming from the sample will also affect downstream experiments. These contaminants are nucleases, proteases, or PCR inhibitors like humic acids included in soil samples or phenol compounds and polysaccharides from plant specimens. The first step for successful microbiome research experiments is therefore selecting optimal tools for unbiased microbial cell lysis. With more than 20 years' experience in developing comprehensive sample preparation solutions, MP Biomedicals offers an efficient method for the preparation of high-quality nucleic acids from challenging sources. The FastPrep system includes several bead-beating homogenizers for thorough homogenization and cell lysis of the most resistant samples. They are used in combination with lysing matrix tubes containing a mix of beads of different material, size, and shape. These lysing matrix tubes are optimized to guarantee a complete homogenization of the sample and the lysis of any microorganism. To provide a complete sample prep solution, ready-to-use purification kits containing the lysing matrix tubes for the grinding step and all the reagents required for the purification of DNA or RNA have been developed. 4 bead-beating homogenizers have been developed to answer the need of specific applications. The FastPrep 24 and FastPrep 24 5G instruments for middle throughput experiments performed in tubes of 2, 4.5, 15 or 50 ml with the option of using a cryogenic sample holder to prevent a temperature increase of the tubes. The FastPrep 96 instrument specifically designed for high-throughput applications. Two 96 well plates can be processed simultaneously, and the mobile SuperFastPrep 2 device, a convenient way to grind samples on-site. FastPrep 24 bead beaters offer a straightforward, powerful sample prep procedure based on the unique figure-8 motion of the instrument, combined with performant lysing matrix particles designed to grind the most resistant samples. 19 interchangeable sample holders are available, allowing a unique flexibility in sample number, size, and temperature control. They are designed for tube size ranging from 0.5 ml tubes to flasks of 250 ml. A sample holder for 96 well plates is dedicated to high-throughput applications. Two categories of sample holders have been designed for either grinding samples at room temperature or in cryogenic conditions. Metal adapters are built for easy decontamination when working with infectious samples. A comprehensive portfolio of lysing matrix tubes composed of 16 different combinations of beads are available in 2, 4.5, 5, 15 and 50 ml, as well as 96 deep well plates to fit any sample type and applications. Lysing matrix tubes are for single use and are certified DNA and RNA free. They ensure a reproducible, thorough lysis of all kinds of cells with no cross-contamination. Once optimal grinding and lysis of any microorganism is ensured, next step will be achieving high-quality metagenomics DNA extraction. MP Biomedicals developed a comprehensive range of complete DNA isolation kits. These kits are split into 3 main groups depending on the purification protocol used to obtain pure DNA. Fast DNA spin kits are based on a protocol purifying DNA with a DNA binding matrix in slurry combined with spin filters used to facilitate the purification process. Spin Easy DNA kits include ready-to-use silica membrane spin columns designed for the purification of DNA. A complete range of specialized sample type specific DNA extraction kits have been developed. MagBeads DNA kits are designed for automated purification of genomic DNA using DNA binding magnetic beads and following a bind-wash elution process. The first kits that MP Biomedicals launched were Fast DNA Spin Kits. Fast DNA Spin Kits include a DNA binding matrix solution supplied in slurry that offers the flexibility to increase its volume when using cell-rich specimens and thus purify high yields of DNA. These kits include as well specialized lysing matrix tubes that guarantee a thorough homogenization of the most resistant samples and the lysis of all the included cells. Pure DNA is obtained in 30 minutes. The Fast DNA Spin Kit for soil belongs to this product line. This kit is designed for the isolation of DNA from environmental and fecal samples, as well as food and plant specimens. It is listed in over 14,000 articles and is described as a gold standard method by several research teams. It ensures an efficient lysis of microorganisms in a few seconds and complete purification process in 30 minutes. This means a huge time saving compared to manual grinding methods. Any cell included in the sample is efficiently lysed, even the most resistant ones. Gram-positive and negative bacteria, fungi, yeast, bacterial and fungal spores, protozoa, viruses, plant and animal cells. High yields of high-quality DNA are purified with this method, with the guarantee of an efficient removal of humic acids and all other PCR inhibitors included in the sample. Strengthened by this success, MP Biomedicals' R&D team developed new kits called SpinEasy DNA kits. SpinEasy kits offer the ultimate of convenience and ease, as the DNA purification process is based on a bind-wash-elute protocol using ready-to-use silica membrane spin columns. These kits include as well specialized lysing matrix tubes for the initial grinding of the sample. Complete DNA isolation is achieved in 30 minutes. The SpinEasy DNA Pro Kit for soil is designed for the isolation of pure microbiome genomic DNA from challenging soil types, including those with low biomass or those highly contaminated. Lysing matrix YB effectively lyses various microbiome populations, including bacteria, fungi, viruses, and protists. New inhibitor removal chemistry removes inhibitors even from highly contaminated samples. DNA is purified using a bind-wash-elute protocol with ready-to-use columns able to bind up to 50 micrograms of DNA. Various soil samples like topsoil containing a high concentration of microorganisms or challenging samples like sand or peat soil were used as a starting sample to purify microbial DNA with the SpinEasy DNA Pro Kit for soil. High yields of pure DNA suitable for downstream applications was obtained with every soil sample type. The SpinEasy DNA Pro Kit for feces uses the same extraction and purification technology. Lysing matrix YB allow for thorough lysis of any microbial cell present in fecal samples. Formulated buffers are designed to get high yields of pure DNA without co-purification of RNA and complete removal of PCR inhibitors. The SpinEasy DNA Pro Kit for feces provides high-quality DNA from various fecal samples. DNA is suitable for PCR, qPCR, and next-generation sequencing, 16S and whole genome, without the need of a further inhibitor removal step. SpinEasy DNA Pro Kits for soil and for feces provide a simple and efficient DNA isolation method from any environmental and fecal samples, even the most challenging ones. Using lysing matrix tubes included in this kit, together with the fast prep bead beater, will ensure a thorough lysis of all microbial cells from the sample of interest. Environmental and stool samples have a high level of PCR inhibitors. This is why specific contaminant removal buffers were optimized to eliminate efficiently humic acids, heavy metals, and aromatic compounds found in these sample types. High-quality DNA purified with SpinEasy kits is then ready for use in downstream metagenomic application. Another SpinEasy kit is specifically dedicated to water samples. It is the SpinEasy DNA kit for water. In this protocol, water is first filtered with the 0.22 micrometer filter membrane included in the kit. Microorganisms from the water samples are retained on the filter. The filter, covered with bacteria and other microbial cells, is then placed in the supplied lysing matrix E tube and processed with the fast prep instrument. All the cells are efficiently lysed and DNA released in the supernatant. DNA is then purified from the supernatant with ready-to-use silica spin columns. Different types of water samples, such as river and seawater or wastewater, were used to isolate DNA with the SpinEasy DNA kit for water. On the table here on the slide, you can see that high yields of pure DNA was extracted for all of them. On gels, you can see that DNA is amplified successfully when performing a PCR on the 16S RNA gene. The SpinEasy DNA kit for water offers performance and convenience in isolating DNA from any kind of water samples. It contains all the required material to perform the extraction, including filter membranes to filter large volumes of water and collect microbial cells. 5 ml lysing matrix tubes, specifically developed for this application, are used to lyse all the microorganisms from the filter in a few seconds. DNA is cleaned up with inhibitor removal buffers and ready-to-use silica columns. Pure DNA is thus ready to use in downstream applications, including NGS. For the study of microbiomes from bodily fluids or milk, for example, or for experiments with bacteria in culture, we recommend the use of the SpinEasy DNA kit for microbiome. Like with other SpinEasy kits, the sample is first added to the lysing matrix tube together with a lysis buffer. The sample is then processed with the fast prep to thoroughly lyse cells. Then, a chemical inhibitor removal step is performed on the homogenate. And finally, after this step, DNA is purified using a bind wash elute protocol with ready-to-use silica spin columns. As you can see on this slide, the SpinEasy DNA kit for microbiome performed particularly well and better than other methods for the isolation of DNA from milk samples. High yields of pure DNA were obtained when using fresh, skim, and whole milk spiked with bacteria. Working with sputum, urine, milk, or a pellet of bacteria, the SpinEasy DNA kit for microbiome offers an optimal and reproducible lysis of microorganisms included in the sample. High-quality DNA, free of inhibitors, is obtained and is ready for use for reliable downstream applications. Host DNA makes up the majority of DNA in bodily fluids like saliva samples, for example. A high host DNA background hinders the application of shotgun metagenomic sequencing in characterizing the microbiota of human and animal specimens. The SpinEasy host depletion microbial DNA kit provides an easy-to-use protocol for the isolation of microbial DNA from samples containing high amounts of host DNA. In the first step of the experiment, a specially formulated buffer is used to selectively lyse eukaryotic host cells. Next step is enzymatic degradation of the released host DNA and collection of bacterial cells. Bacteria are then lysed through a combination of microbial DNA extraction buffers and highly efficient homogenization using fast prep. DNA is purified using a convenient silica membrane spin column technology, including 2 wash steps. A PCR performed on DNA isolated from saliva without host depletion and with the SpinEasy host depletion microbial DNA kit using human beta-globin primers and 16S bacterial RNA primers pointed out that host DNA was efficiently deleted with the SpinEasy host depletion microbial DNA kit protocol. The SpinEasy host depletion microbial DNA kit offers the benefit of effectively eliminating host DNA cells. Host DNA can be reduced from 95% of DNA in non-depleted samples to only 1% host DNA when purifying DNA with the SpinEasy host depletion kit. This complete kit includes lysing matrix tubes to ensure the lysis of all the microbial cells from the sample, even the most resistant ones. Highly pure bacterial DNA is ready for microbiome profiling studies. Another benefit of host depletion is the cost of sequencing, as samples contain almost only bacterial DNA. To assess the functional activity of a microbiome, MP Biomedicals' next objective was to obtain pure RNA for metatranscriptomic analysis. For the isolation of RNA from soil and other environmental samples, two kits are available. The Fast RNA Pro Soil Direct kit, based on a technology using the whole soil specimen as a starting sample, and the Fast RNA Pro Soil Indirect kit, based on a protocol including a preliminary washing step and a filtration of the soil sample. This step allows the collection of a microbial pellet that is used as starting sample for the RNA isolation process. With both kits, grinding and cell lysis is performed with lysing matrix E tubes and the FastPrep instrument. RNA is isolated with a phenol chloroform extraction protocol. Removal of humic acids inhibitors and purification of the RNA is achieved by selective binding of the RNA to the RNA binding matrix. A second level of purification is performed by filtration of the RNA through quick clean spin filters. Fast RNA Pro Soil kits provide high-quality total microbial RNA. RNA degradation is prevented using protective reagents during the whole extraction and purification process. Microorganisms present in environmental samples are all effectively lysed with the help of lysing matrix E tubes and the FastPrep instrument. RNA of the highest quality, free of PCR inhibitors, is obtained ready to use for downstream applications. For stool metatranscriptome studies, the SpinEasy RNA kit for feces provides a protocol for purifying pure RNA from fecal samples. Excellent cell lysis is achieved in just a few seconds by bead beating with lysing matrix YB tubes. Specially formulated buffers allow to get rid of all contaminants and PCR inhibitors. Selective binding of the RNA to SpinEasy ready-to-use columns is used to purify RNA and get premium RNA quality. Tested with many different human and animal fecal samples, the SpinEasy RNA kit for feces delivers reliably high yields of pure RNA without detectable DNA contamination in less than 20 minutes. Similarly, the SpinEasy RNA kit for bacteria is designed for the isolation of total RNA from gram-positive and gram-negative bacteria, as well as bacterial spores. It includes lysing matrix B tubes that will ensure a reliable lysis of any strain of bacteria. Using an RNA protective extraction buffer and a simple bind-wash-elute workflow, high yields of pure RNA is obtained within 30 minutes. RNA is eluted in pure water and is ready to use in downstream applications. Escherichia coli and Staphylococcus aureus bacteria pellets were used to isolate RNA with the SpinEasy RNA kit for bacteria. Extracted RNA were of high yield, quality, and integrity, as indicated by concentration that were obtained and OD ratio that were around 2. As you can see on the gel pictures, purified RNA were successfully amplified by RT-PCR. The microbial composition of environmental and fecal samples varies from one sample to another, and the amount of the available sample is sometimes limited. moreover simultaneously investigating the taxonomic composition, gene contents, and gene expression patterns of microbial communities provides valuable information. Our next goal was therefore to optimize protocols for co-extracting DNA and RNA to enable paired metagenomics and metatranscriptomics sequencing. The SpinEasy DNA-RNA kit for soil provides an easy-to-use workflow for simultaneous isolation of pure microbiome genomic DNA and RNA from challenging soil types, including those with low biomass or those that are highly contaminated. Samples are first ground by bead beating with lysing matrix YB to lyse efficiently all the microbial cells. The homogenate is then incubated with an inhibitor removal buffer used to remove efficiently contaminants from environmental samples. Following this step, the supernatant is collected to separate and purify DNA and RNA by using respective silica ready-to-use columns. The isolated DNA and RNA shows no contaminants and is immediately ready to use in downstream applications, including qPCR and RT-qPCR without the need of further inhibitor removal steps. Experiments performed with various soil samples pointed out that DNA and RNA extracted with the SpinEasy DNA-RNA kit for soil show high yields and integrity across high biomass, high contaminant, and low biomass soil samples. Using the same technology, the SpinEasy DNA-RNA kit for feces provides an easy-to-use protocol for simultaneous extraction of microbial DNA and RNA from the same stool sample. The workflow is very similar, with three main steps. First, sample grinding of up to 250 mg fecal samples with the fast prep and lysing matrix YB tubes. Second step, PCR inhibitor removal with a buffer specially developed for eliminating contaminants from feces. And the third step, separation and purification of DNA and RNA by using respective silica ready-to-use columns. Extracted DNA and RNA are eluted in pure water and are ready for use in any downstream applications. Tested with many different animal fecal samples, the SpinEasy DNA-RNA kit for feces extracted DNA and RNA with high yields, optimal OD 260-280 and 260-230 ratios, as well as high integrity. You can see on this slide RNA samples extracted from horse, duck, and pig fecal samples that were loaded on an agarose gel. No smeared nucleic acid corresponding to DNA contamination could be observed in RNA samples extracted with the SpinEasy DNA-RNA kit for feces. After this development of a complete range of kits for manual extraction of DNA and RNA, our next objective was to optimize solutions for automated extraction dedicated to microbiome research applications. Two automated nucleic acid extraction platforms have been recently added to MP Biomedicals' molecular biology product portfolio. The MPure 32 instrument that is designed to process up to 32 samples simultaneously, and the MPure 96 instrument that is designed for high-throughput applications and purifies DNA and RNA from up to 96 samples in one go. On this slide you can see the new MPure 32 instrument. It is one of the smallest automated systems on the market, 38 per 35 cm on the benchtop. It is easy to use thanks to the touchscreen with user-friendly icons containing up to 100 preload programs. Ultra-pure DNA and RNA is isolated from 32 samples in less than 1 hour in a reproducible way without the intervention of the user. 2 heating blocks ensure the assay to be run at the optimal temperature for maximum yield and result consistency. After use, the built-in UV light decontaminates the instrument. The MP 96 instrument is a fully automated platform handling magnetic beads for the purification of DNA and RNA of up to 96 samples in one run. An intuitive interface makes it user-friendly. Its specialized spin tips enable supermixing efficiency of magnetic beads and larger processing volume of up to 1,600 microliters. The MP 96 is a flexible platform. It can be configured with your own protocols for any specific application or experimental requirements. MP Biomedicals' R&D team optimized a complete portfolio of magnetic bead-based kits for DNA and RNA purification from a wide range of samples. MagBeads fast DNA kits include magnetic beads with high binding capacity and selectivity for genomic DNA. Specialized lysing matrix tubes are added to these kits for a complete and efficient grinding of the sample and cell lysis before proceeding with the purification of the DNA. The MagBeads Fast DNA Kit for Soil is designed for the purification of DNA from soil and other environmental samples. The use of high DNA binding magnetic beads and extraction buffers specially developed to eliminate humic acids and other PCR inhibitors allows to get high yields of high-quality DNA from soil, sediment, sludge, compost, or manure. Extraction is performed with one of the MPO instruments or any other automated purification instrument in a process that takes 1 hour. Purified DNA is ready for use in downstream applications including NGS experiments. This slide shows that the Magnetic Beads Fast DNA Kit for Soil achieved better performance across various types of soil samples when compared to similar other DNA extraction kits. MagBeads technology, together with the FastPrep homogenizer, guaranteed the extraction of high DNA yields. The unique formulated inhibitor removal solution ensures a high level of purity and increased OD 260-230 ratio. Similarly, the MagBeads Fast DNA Kit for Feces allows the isolation and purification of high yields of DNA from human and animal fecal samples. The protocol starts with the grinding step of the stool sample by bead beating with the lysing matrix tubes and lysis buffer included in the kit in combination with the FastPrep instrument. After centrifugation to pellet lysing matrix beads and cell debris, the supernatant is collected and the purification of the DNA is performed by a bind, wash, elute process using magnetic beads. This process is performed with the MP 32 or MP 96 instrument or any similar instrument. This slide shows results obtained with chicken fecal samples. High yields of high-quality DNA was isolated with the MagBeads Fast DNA Kit for Feces. DNA was suitable for use in downstream applications without any additional purification step. To offer the ultimate inconvenience, MP Biomedicals produces MagBeads kits composed of 96 well plates pre-filled with reagents and ready for use with the MP 32 and the MP 96 instrument. This leads us to conclude that MP Biomedicals offers a complete solution for microbiome research sample preparation. To answer the need of any research project, the comprehensive portfolio allows to choose the right fast prep homogenizer with the appropriate lysing matrix and the sample-specific DNA and RNA extraction and purification kit. For high-throughput applications, combining an MP automated platform with ready-to-use DNA and RNA purification kits is a convenient and time-saving option. MP Biomedicals is pleased to offer you free sample kits of fast DNA spin kits, Spin Easy and MagBeads kits to give you the opportunity to test these extraction and purification protocols with your starting sample. A demonstration of the fast prep and MP 32 instrument can also be organized in your lab. For more technical information, detailed brochures, application notes, and videos are available on request. Thank you for your attention.